Welcome to this episode of Eureka. Imagine when we are born, or rather when you are conceived to be more precise, a sperm from the father and an egg from the mother fertilizes and first cell is born. Then the first cell divides into two. Then each of them divides into two, that means it becomes 4. The 4 becomes 16, 16 becomes 32, and when we are born, we have approximately 50 lakh crore cells. All of them are almost same in terms of DNA inside them, protoplasm inside them, cell machinery inside them. But then some of them go on to become our head, some of them go on to become our finger. How does a cell know that I have to become the finger? How does a cell know that I have to become the ear? This is a question that we are going to explore with today's guest, Prof L S Shashidhara, Head of the Department of Life Sciences, Indian Institute of Science Education and Research, Pune. Thank you, sir. Thank you, sir, being with us. Before we proceed with the conversation, let's take a quick look at his brief profile.
Professor L S Shashidhara is a well-known geneticist and developmental biologist. He is presently heading the biological department in IISER, Pune. Hailing from Karnataka, Shashidhara has researched on evolution and the functional genomics of appendages using the fruit fly as a model system. His studies assisted in a wider understanding of the relation between genes and diseases in human beings, and also in the development of cancer drug discovery systems. Professor Shashidhara was awarded his PhD from Cambridge University and has a wide teaching experience in various esteemed institutions. He has received various awards for his outstanding work, noted among them being the Shanti Swarup Bhatnagar Award.
Thank you for being with us in this show. That's a question that we are all kind of intrigued. You know, there is a cell, the cell divides, cells are same. How does a cell know that I have to become a head? How does a cell know that I have to become a finger?
Yeah, so I guess this is the fundamental question in science, not just in biology, because finally you want to understand yourself, right? So the science is all about understanding the natural mechanism and leading to self as well as the non-self, right? Whether you study gravity, whether you study origin of life, whether you study consciousness, finally it all boils down to asking the very fundamental questions. And development of an organism is one of the fundamental questions, because the so-called the question of self comes from the very first event, starting from fertilization and then the subsequent development to a whole organism. And you just mentioned that we are made up of something like close to about 50 trillion cells. That number varies, 50 to 100 trillion, because there are a large number of cells are continuously dividing and supplementing for worn and dying cells because of wear and tear in the tissues. So it may, it's very difficult to calculate how many cells we have. Let's say about 50 trillion cells. Now assume, it's just complex, how complex the problem is. 50 trillion cells should know that what they are, what they are supposed to do, where they are supposed to get, be resident in the body. And in the top of all these things, there should be coordination amongst themselves. For example, if I lift my hand, right, all the billions and billions of cells in my hand should go up and down at the command of the brain, right? So this kind of a coordination is also important. So it's not just a mass of cells is produced and each cell is assigned some function. That can't happen, because that should be a coordination among these cells during the birth itself, right? The whole developmental processes. So in nature, obviously, simple systems lead to complex system. You cannot expect complex system to emerge, overnight or in one single step, right? So in a way, it's step by step increase in the complexity. And it's actually the use of this kind of model organism, which I work on, Drosophila, which has contributed most to this understanding of early developmental events, which starting from a single simple system, how you get multicellular complex systems, right? So many times we ask the question, does the cell know what it is, right? So you can ask the same question to even a single cell system, the embryo itself, the zygote we call in the technical term, and then when it becomes a multicellular system, then we call it as an embryo, right? The fertilized egg or the zygote, does it know the two ends of it, right? Now, what is the end for it, right? Whether the this end, head end, or tail end, because only one cell. There is nothing like head and tail, right? So apparently, even the cells have their own polarity.
Okay.
Right. That means
just
that
like a magnet has a north pole and south
exactly,
pole. Okay,
exactly. In fact, you can call it as plus and minus end.
Okay.
Right. A cell too has a plus and a minus end. There are internal asymmetry according to this two ends.
Okay.
But when the first cell is when you look at the egg even before fertilization, at a particular stage during egg maturation, there is actually no polarity. There is nothing like plus and minus end. Both ends can be minus end. Both ends can be plus end kind of a system. But it so happens that both ends are minus end. There is no plus end in the two ends. So because of this, you don't expect any polarity. If there is polarity, you cannot see any asymmetry. Without asymmetry, you cannot generate a body like ours, right? There is an asymmetry. There is a head on one side and the opposite on the other side. Now, at some point during development, the cell assumes polarity because of some interaction with the cells of the mother, and that basically, in a way, what we are trying to do is, the information is already there in the mother, which is the head side and which is the tail side. That information need to be transferred to the egg, so that the next generation, when it comes out, it should know where the head and the tail is. Okay. Right? That's precisely what the converting a complex problem to a simple question.
Okay.
It's still a research question. I'm not saying it's an answer, but it's a way of asking the question. Now, again, the next question is, as you already mentioned, all the cells have the same genome. That means genetic material, the DNA, which is present inside the cell, is exactly the same. Now, from this similarity, how do you generate diversity, right? And if you, for example, even today, if I take different cells from my body, you see that the genetic material is exactly the same.
But on the other hand, the skin cell is quite different from, for example, the cell in my eye.
Cell in your eye. Right? Under microscope, for example, if you look at the neuronal cell or a muscle cell, they look as if one is a dinosaur and another is, a whale, right? So in a way, you see them as so different, almost like how can you even imagine that they come and belong to the same organism? Forget about species. Right? It's the same organism. They belong to the same organism. So the way it happens is the genetic information is interpreted in slightly different ways in different cells.
Okay.
Now, that means the very first step of a polarized embryo or a cell, what needs to be done is interpreting the genome or genetic information differently in different parts of this embryo. And that's what the developmental mechanism means at the molecular level, at the cellular level. Reinterpretation of the genetic information at every stage during development. So again, this also has to have a beginning. Right? And this beginning has to have a simple beginning. You cannot expect again a very complex beginning. It's almost like imagining, let's say there are about 1 billion people of country, okay, at 12 o'clock in the midnight, asking them to think exactly the same thing, right? It's impossible, right? Now, obviously, you cannot generate 1 trillion cells or 50 trillion cells and asking them to start thinking that you become a neuron, I become a muscle, I become a muscle of the finger, and another one become a muscle of the thumb and or a leg or something, right? So this has to be done in a very sequential way, in a hierarchical way too, right? Now, how do you generate complexity from a single system, simple system, right? A single protein now has to generate complexity. Here is where a very elegant system has evolved over several million years of evolution. Is this protein, when it is expressed in a concentration gradient.
Okay.
Depending on how much of this protein is present, different information is generated. Right? The same protein at highest concentration makes the head. A slightly moderate concentration, it makes the trunk, which is basically the so-called the chest region of our body, and least concentration or no protein may lead to abdomen. Right? So you think of it, single protein now can generate 3 or 4 different types of body parts.
Just because of its concentration.
Okay. Now you think of 3 or 4 such kind of proteins involved in this. Automatically now you can think that there are about 30-40 different parts of the body, right?
Yeah.
So our body, a human body, can be divided into about 33 different compartments. Okay. If you take an X-ray film of your backbone, you see 33 vertebrae, right? There's a ladder-like structure. And each unit represents one part of the body, and that's how the development happens, because the nerves are connected to that one. Okay. So, for example, all the nerves coming from my hand, they go through the arm and then go through a particular vertebra, and from there it goes to the central nervous system.
Okay.
So that's how we generate.
So this 33 parts roughly is generated because of 33 different kinds of
Not really
concentration.
33, a smaller number. Certain permutation combinations of different proteins expressed in a concentration gradients, they lead to formation of 33 diverse body parts.
Very interesting. We'll continue this conversation, but we need to take a very small break. Don't go away. We are going to have more interesting questions. How does the body know that it should produce only 5 fingers? Why it is not producing 10 fingers in the same palm? These are all questions that one can ask, and our guest today has worked on this area. We'll ask this question, we'll converse on this issue after the break. Don't go away. Welcome back to Eureka, and we are having a very interesting conversation with Prof L S Shashidhara, who is the head of the Department of Life Sciences, IISER, Pune. Before the break, we were discussing about how each cells in our body knows where they are and what they should be doing, and then we have been discussing that. And before the break, I was pointing out to the audience that how does the body know that it should produce just only 5 fingers and not, let's say, 10 fingers from the same palm? You have worked on that area. Can you elaborate on that?
Well, I have not directly worked on that area. Basically, like I told you, I work on an insect called Drosophila. Drosophila is also a multicellular organism, but it's an insect, right? It's not even a vertebrate. And I specifically work on how the forearms are different compared to our legs, right? See, for example, when humans started walking on 2 legs, basically we got 2 arms which are free, which can be done for other purposes, right? For example, compared to other organisms like cattle, for example, they had to walk on 4 legs, right? They don't have any other arms, right? But the 4 legs, the 2 in the front, 2 in the back, they are more or less similar. But look at some other organisms.
For a cattle, the front leg and back legs are almost same.
Are the same, but used exactly for the same purposes, like walking and running. The same thing for horses or any other 4-legged animals. But look at birds. The birds, there has been a modification. Now there are 2 legs, but they are at the 2 of the so-called the rear legs. The back legs are still legs, are used for walking, whereas the 2 front legs are modified into wings. So now they can fly. Similarly, in human, the 2 front legs are now modified into arms, which we can use it for other purposes. That's why we are more sophisticated in use of tools compared to a 4-legged animals, right? So my question is related to understanding how the number of appendages are determined in the body, right? Why there are only 4 legs, why there are 2 arms and 2 legs, or 2 wings and 2 legs in birds. Similarly, in insect, there are multiple different types of one. For example, butterflies have 4 wings, whereas houseflies like mosquitoes and houseflies and all, they have only 2 wings. So how these numbers are determined? The mechanism is dependent on a particular family of proteins called Hox proteins, on which I work on. And it's the
Hox
same
proteins,
Hox proteins. Yeah? They are basically, for a variety of reasons, they are called Hox proteins. Now, the same proteins are also involved in variety of other aspect of our body formation, right, during development. So the question that you asked about, why there are 5 fingers, why there are more number 5 fingers. So there are multiple reasons for this. One reason is the Hox protein is the one which help in determining how many fingers should be there, right? So for example, you have seen people with polydactyly. Polydactyly is a phenomenon where there are more than 5 fingers. There is a 6 fingers. Some people have even 7 fingers in the same palm. And those people will have this Hox protein expression is slightly different. So instead of restricting to the 5, they have more Hox protein expressed and leading to more fingers, right? There are fixed amount of Hox protein should be expressed in this one, and changing in the levels of Hox protein leads to increase or decrease in the number.
It's very interesting. I mean, before I came for this interview, I was telling my friend that I'm going to interview you, head of the Department of Biology. He sort of scratched it and said that, oh, you are going to talk about unpronounceable Latin names. When you are in school, biology means remembering a lot of Latin names. But I'm talking to you about something quite different. What is biology today?
The origin of universe 15 billion years ago, it was only energy. Now there are different types of particles emerged out of this energy because of some dynamics, and we are all conforming to certain laws of physics. The same one also led to chemical world, what we call as carbon dioxide, oxygen, hydrogen, or the individual elements to small compounds. right? Molecules like carbon dioxide or other things, right? Water, H2O, which is very important for Earth, for life. From that one, somehow 3 billion years ago, life emerged out, right? That origin of life has basically is dependent on the chemical world, which was dependent on the physical world. That's why even the organisms or biological world too conform to the laws of physics. There is no violation of laws of physics. It is actually many people think that biological world violates the laws of physics, which is not true, right? Now, today's biology, learning as well as research is about how this world has come about out of a chemical world, right? And how it is diversifying. The much of biology is to understand diversification rather than trying to see, how do we perform on one particular task, right? Look at it, millions and millions species are around. Look at the people, there are billions of people and each one look differently, each one respond to environment differently. And if you look at the way our neurons function, we also respond to the similar kind of stimulus differently, right? So this is what the biology is.
This was very interesting. Another interesting thing, when I was walking into your department, this life sciences department, in few halls, I saw mathematics. Again, for a student, you take biology group, means you don't take mathematics. But here you are having a life sciences department where people are conversing in mathematics. What is this? I mean, again, I'm sure many of us would be zapped.
Thanks for asking this question. It's a very important question. This is exactly what we want to do in IISER. We want to change this perception because the biology is basically dealing with, as I told you, with diversity. And there are the numbers we have to deal with is mind-boggling. The human body is made up of 50 trillion cells. That's how we started this conversation. Now, if you're talking about very different interactions, right? And also I mentioned to you that the concentration of a protein is more important than the presence and absence of a protein, right? If that's the case, basically biological phenomena has to be looked at from more quantitative perception rather than a very qualitative way, right? And also it's a continuum. Chemical world to biological world, within the biological world, bacteria to human, it's a continuum. In this continuum, you cannot but look at each step in a very quantitative way, right? So quantitative biology is the order of the day, and to understand the mechanism of anything, you need to look at it more quantitatively. So without mathematics, you cannot think of biology these days.
So if you are going to be a biologist in today's world,
yeah, without mathematics, we would be not doing a good justice to do biology research.
That's very interesting. You were talking about this is a new model that you are going to create in IISER. So we'll discuss this after a break. After the break, we are going to discuss about what is this new model in IISER, Indian Institute of Science Education and Research. Let's have a conversation. We'll take a small break. Don't go away. Welcome back to this episode of Eureka. We are having a very wonderful conversation with Professor L S Shashidhara, who is the head of the Department of Life Sciences in Indian Institute of Science Education and Research, Pune. How did you choose this research? I mean, kind of in our times when we are, leaving school, the dream would be to become a doctor or an engineer, or perhaps write some competitive exam and get into civil service. What made you come into this?
I did all those things. Oh. I also wrote competitive examinations, and I wanted to go to a professional course. I chose agriculture over engineering or medicine.
Okay.
Well, I can also say that, to be honest, I didn't get the medicine, and I don't think I managed well. So I did have an option of going to either engineering or agriculture. There are three or four you can say professional courses of those days, which are very attractive: engineering, medicine, veterinary, or agriculture. I chose agriculture, and I got fascinated because of the quantitative aspect of the agriculture research, particularly genetics. Okay. Genetics is very mathematical, right, compared to other aspects of biology. So for me, genetics was really I hooked onto it. And fortunately for me, they had fantastic teachers teaching genetics, plus at the same time they also took me to the world of evolution. And having read Darwin and post-Darwin work on evolution, I sort of hooked on to understanding the evolutionary aspect of biology, and that's where I made a shift from plant genetics to animal genetics, and we started using Drosophila as a model organism to study very different aspects of biology, particularly developmental biology.
Yeah,
so that's how I started.
So then where did your career path
So I went to PhD. I went to Cambridge to do my PhD, and then after my PhD, the best place to do good research in India in those days in the area of genetics or developmental biology was TIFR, Tata Institute of Fundamental Research. I ended up going there. From there, I then went to CCMB because CCMB wanted to start some new area in that time. So they invited me, and I started my career in a much bigger way at CCMB, where I spent more than 12 years. And then that's the time, you mentioned about IISER a little bit just a while ago. See, one of the problems of education system is we have separated undergraduate education from research. So most of our undergraduate education happens in colleges where there are teaching only institutes, right? Teachers themselves do not do research. And professional courses are being taught in some places, like in medical colleges where some medical research may be happening, engineering colleges or IITs where some research is happening. Again, engineering college themselves do not do research. So they're all teaching only institutes. This actually basically means you are transferring knowledge without telling them how the knowledge is produced.
But then if you look at the way most people think, for example, if you want to do research, then you join a research institution. If you want to do teaching, then you join an educational institution. But here you are saying that researchers should do teaching. Exactly. Would it not actually kind of hamper the research?
No, it's not. So the two reasons. One is, let's say starting from the societal importance. Now what is education is for? Education is not to remember these so-called Latin names of very different organisms or names of different particles that exist in nature, right? The education is to solve the problems of life. Right? Problems of your own personal life or problems of the society. For example, you may become an IAS officer, you may become bank manager, you may have to deal with the societal problems. Then you have to, when you solve problems, it is almost like a research. What we do in research, we solve problems, but using scientific methods. Advantage of using scientific methods to solve problems is you can be very, very precise, right? And you can actually identify ideal solutions. Now, this problem solving ability which is there in the researcher is never transferred to the students in masses. You may transfer to some PhD student, but not in a very large number. In a way, it's actually injustice for having acquired so much of research skills at the cost of public money not to transfer that to the other people, right? So one is, in that context, research and teaching should be integrated. Advantage for researchers themselves is, when I teach, I get lots of interesting ideas to do my research based on the questions the students ask.
Because the students have a fresh mind, they don't have any inhibition, they can ask what is called as stupid questions.
They know, they ask very creative and innovative questions, and that will help my research.
Yeah, very interesting. Another issue that is bothering the country today is the issue of higher education, where you are talking that we need a fresh look at how we are going to do about it. The whole debate about public funding for research, public funding for basic research, how does it matter how we know, for example, which part of the cells are going to become the head and which is going to become a hand? Are there not more important problems of the society? People are suffering from diseases. These are the kind of questions that is asked in public arena. How would you respond to this kind of concern?
What we do in basic science is, because the basic science asks questions for which absolutely no answer is available, you basically start developing research methodology to solve those problems, right? And in the process, you develop capabilities in the trained people to solve problems. This is very important to solve the problems of the society. Otherwise, I'll tell you what, one of the problems in India facing is, someone may say that, so what, Germany makes some technology develops, or America technology develop, maybe technology, we can just import it, we can just buy it. We have money, we can buy it. Why you need to fund the basic science? Even to have an imported technology, you need to first negotiate what technology we need, right? If you don't have internal capability to negotiate, they can tell us for a ride, right? For example, and also you need to be scientifically literate to even deploy the technology. There are multiple examples. I can give you some examples. You can't even imagine that the basic science has helped certain kinds of industry, right? Like basmati rice export, it's largely has helped because of some basic science that is done in the DNA fingerprinting lab, right? Or in the Surat, the Surat has now out competed the Antwerp in diamond polishing, right, and cutting and polishing, and that's because it started using lasers at the same time with the low labor cost, right, and the lasers we could use in India extensively because we are all literate in physics, starting from Raman, right. Raman never thought of Raman did work on diamonds, by the way, but he was not thinking of cutting and polishing diamonds.
So this is a strength in the basic sciences which actually provides us scope for last, because we don't have much time in this conversation, but then one last question. What you think is a major challenge for higher education in India?
The major challenge right now is how to make students think innovatively. See, we are giving too much importance to acquiring certain knowledge and writing an examination so that you can pass and get a degree. Actually, we don't need any of this. So that basically analytical skills and problem solving ability is what we need to inculcate among students. So we need changing the pedagogy is the major challenge in higher education. But pedagogy depends on intense interaction between teacher and student. And with now the challenge comes. Now our gross enrollment ratio in higher education increasing because of better economic situation in the country, but we don't have teachers. So how to give good quality education with less number of teachers to several hundred more number of students that we could, that were coming to the colleges just 10 years ago.
That's an important challenge. So we have two major challenges with regard to youth and higher education in India. One is, how do we change the curriculum and syllabus so that it becomes more innovative and trains people in problem solving and not just remembering some facts and figures? And second, how are we going to meet the demand of rising generation, more number of young people coming for higher education? These are two major challenges for higher education, and we had a very wonderful conversation with Prof L S Shashidhara. Thank you for your time and sparing your time. Next episode, next week, we'll meet and have another very interesting conversation.